Good afternoon, everyone. This is Paul Eniver, and welcome to our webinar today. Um, just by way of introduction, so I work for Canapa US, which is an asset management and an advisory firm, where I oversee the healthcare unit of Canapa US, which advises the Canapa Advanced Healthcare uh, fund, and it's a fund which invests broadly in the healthcare space, but with focus on diagnostics and medical devices. And in that capacity, I also have the privilege of serving on the board of directors of GNOME. So very excited to be here today to introduce Dr. Crawford, who's going to be presenting in today's webinar. Dr. Crawford has prepared a talk for us around some of the business elements of genomic testing and his work in terms of surveying early adopters. Dr. Crawford actually has quite an extensive resume. He's currently the Executive Director and Senior Vice President of Laboratory Services at North Shore Long Island Jewish Health System, which is obviously an academic role as well as a business role, where he's also the chair of the Department of Pathology and Laboratory Medicine at the School of Medicine, where he has academic responsibilities as well. So it gives him a unique perspective on both the business and scientific sides, clinical aspects around genomic testing. Prior to being at North Shore Long Island Jewish, where he's had that role for 6 years, he was 10 years at the University of Florida in Gainesville in a similar role. Over the course of his career, he's been widely published in a number of different areas and has extensive background in all elements of pathology and diagnostics as well. So, Dr. Crawford, thank you very much for joining us today. I'll hand it over to you to go forward with your presentation.
Thank you very much, Paul. I was invited to give this talk on the basis of a paper that a study group, a task force of the College of American Pathologists published in July of this year, and I will get to it in just a moment. I think it's important to give a disclaimer to what Paul just said in introduction. I'm a GI pathologist by training, and I'm not a molecular pathologist. Rather, if you look at these words that are on the title slide here, I do carry responsibility for decision-making, at least at the laboratory level, for implementation of technologies in our health system, which is a 16-hospital health system and sees about 16,000 new cancer patients per year, responsible for about 30,000 live deliveries in the greater New York metropolitan area. So whether or not genomic testing comes into our health system is an intensely important question for me. And in this survey of early adopters, I was not one of the people surveyed. Rather, I was chair of the task force, and so what I'm presenting to you is the learning that came from that, and I can tell you right off the bat, I am traveling this own journey myself. If you have questions during the course of the seminar, and I will try to manage my time so that we can deal with questions and answer at the end, on your bar there is a question box which you can type in a question. The other thing in front of you is that you have a chat message area, so if there are any issues with my speaking or the transmission, you can contact the moderators to the chat box, but for questions, we'll tee the questions up for the end of the talk, and I look forward to getting to that part. The disclaimer or disclosure is that I'm Vice Chair of the Managing Committee for a Clinical Trials Research Organization in the New York area. It has no relationship to this particular talk. So the business working group of the College of American Pathologists Personalized Healthcare Committee was commissioned at the end of 2011 to try to address the question of why do genomic testing from a business standpoint. This was not a scientific question. Other task groups of the Personalized Healthcare Committee were working with that. But I raise my hand, volunteer. Let's see if we can figure out the business arguments for bringing genomic testing into a health system. And shown here are the members of this group. I do want to note that Jeff Kant, University of Pittsburgh, succumbed to a terminal illness during the course of this, and we miss him greatly. The paper was published in 2014 in Genetics and Medicine back in July, and this is what I will be presenting to you. First, let's define genomic medicine. Precision medicine is another term for it if you go into the literature. For the purposes of this talk, it's defined as massively parallel sequencing to obtain genetic sequence, familiarly called next-gen. I'm not reporting on using high-density SNP, single nucleotide polymorphism microarrays for screening and deletion. This is a talk about next-gen sequencing. And if you look at the clinical applications in the massively growing literature for next-gen sequencing since 2005, when it really came into being, medical genetics, which is the somatic genome, cancer genomics, and pharmacogenomics, which is a combination of both the your native genome as well as that of the cancer that might be being treated, and then infectious disease diagnostics. Interestingly enough, in our survey, we were focusing just on the medical genetics, cancer genomics. In our early adapters, there was very little discussion of pharmacogenomics or infectious disease diagnostics. For me, right from the get-go of this task force, I was and continue to be concerned about the fact that in cancer genomics and in pharmacogenomics, the users at the time, this being 2012, 2013, turned to next-gen sequencing when patients were in extremis. In other words, after they'd exhausted all prior therapies, they had failed therapies, or their cancer had changed its genome and was no longer responsive to something it had previously been responsive to. And so these were really desperation measures. And the literature then, and quite honestly through to the present, remains that of dramatic stories of patients who were rescued from the abyss when next-gen sequencing was implemented. That also has examples from medical genetics. For me, a driving question through this whole discussion is, should we be doing testing up front, ab initio? Is it better both for individual patients as well as the population we serve to do next-gen sequencing right out of the gate, just look at it as a lab test, get as much information as you can, and run with it? The next question is by whom, and you'll see this come out in the course of the talk. This is a study of the decision to bring genomics into an institution. The definition of a genomic laboratory, I should confine who we are asking. We intentionally did not reach out to the commercial laboratories that offer genomic testing. I consider those entities, which are for the most part major national laboratories, to have an intrinsic business argument for both putting these technologies on the market and for running them, and certainly Wall Street would agree. Rather, this was a question of, should laboratories in health systems or as independent small market providers be doing next-gen sequencing? The private, so the genomic laboratories per se, we excluded because there's an inherent business argument. For private pathology practices, when we looked at who might be early adopters, they weren't there. There was no one to ask. So really, we found ourselves in reaching out to early adopters that we were looking at integrated health systems, and almost exclusively these were academic. So at the time of this study, which was the summer of 2012, these were the providers of precision medicine, namely the integrated academic health systems. We selected participants by these criteria, having first, they were members or had served on the CAP Personalized Healthcare Committee, or they had a CLIA license to perform next-gen testing, and there were very limited number of laboratories that were doing this. They had a record of peer-reviewed publications in this area, and that was basically my going through the literature to see who was doing next-gen testing and what their institution was, and then reaching out to the laboratory director of that institution, or, and you may all have seen these things as well, dramatic institutional announcements in the lay press of a major institution doing next-gen implementation. Interestingly enough, by this criteria, there were 13 people only serving as laboratory directors, and they all agreed to participate in the survey. The survey was a standardized one-hour interview by one interviewer. That was Jill Kaufman of the CAP. And she asked 10 open-ended questions and pursued responses to those questions as needed. The topics addressed in this survey were: what genomic tests were you choosing to use? In other words, what was the clinical application? What were the drivers and barriers? How did you assess and deal with reimbursement considerations? And mind you, this was 2012, and that landscape has been changing rapidly through this whole period of time. Lessons learned. And the last question, which was actually, I think it was question 10, was how can the College of American Pathologists help you? What sort of resources can we bring to bear? The 38,000 words of transcript were typed out, redacted to anonymize them, put in front of the committee, and then we had to figure out how to analyze them. And if you're wondering why it took from 2012 to 2014 to publish this, it was my wrestling with the issue of how do we do something more than just sort of provide text comments? How do we actually deal with this in a rigorous fashion? I was gratified to find that the method that I came up with consultation with the committee, actually is a standard sociological mechanism for analyzing open-ended interviews, and it's called qualitative response analysis, where basically you parse the text into responses to each core question, identify specific units of measure in that question. In other words, what vocabulary and concepts are the respondents using in any given question, and going through the entire 38,000 words of redacted transcript, there were basically 94 concepts that were in play. I might add, the vocabulary was way beyond that, but a key element of boiling it down to 94 is to develop a lexicon of common terms. Having done that, group these terms into categories, compile participants' use both of the units, and then analyze on the basis of categories. So in essence, what you're trying to distill out is what are the concepts that are in play in response to each question. In the end, 13 institutions interviewed, 12 of them in the United States, 10 different states, and then 1 Canadian province. The interviewees had 93 publications relative to genes and diagnosis in the 24 months prior. And for this set of 13 early adopters, the applications that they intended were involved next-gen sequencing, targeted sequencing. There was quite a bit of discussion of bioinformatics, and also delving into analysis, the molecular analysis of surgical material. Only 2 of these 13 at the time had CLIA licenses to perform this. So here are the survey questions. Why did your institution decide to adopt next-gen sequencing? How did you implement it? What clinical applications? How did you decide between bringing it in-house or sending it out? And I'll tell you right now, 12 of the 13 decided to bring it out. The 13th decided to send it out. How did you fund this? How do you plan on getting paid? Do you consider next-gen service as a competitive advantage? What lessons can you share, and how do you measure successful outcomes? And then, as I mentioned, the last question was, how can the CAP help you? So this is my favorite chart, which is how many words do these people say to us? So here are 13 different institutions. And you can see that some people were rather laconic in their responses and others just kept going and going. I can tell you that the information content between the 1,200 and 1,300 word respondents and the 6,000 was about the same. There was no statistical significant difference between the information content and the number of words said. If we look at the information content per question, perhaps it's natural that in an open-ended interview, you're going to get the most verbiage in response to the first 2 questions. But be that as it may, we certainly did get the most information content out of question 1, which is, why did you choose to do this? and then a lesser amount out of how. And what I'm showing here is just a scatter plot of the number of words per respondent. It gets more interesting when you look at the subsequent questions, because applications was easily answered, cancer or somatic testing. To insource or outsource, again, easily answered. Two of the respondents went on at some length. This is interesting. Here we are trying to find out what a business argument is for bringing in next-gen sequencing, and our respondents for question 5 really didn't have a whole lot to say. There was a broad spread of information content for their thinking about how to engage payers, and a reasonable amount of information content, on what they felt to be their competitive advantage. You can see one institution had a lot to say about lessons learned. The others were fairly concise. This to me is the most interesting. How are you going to measure your outcomes? And for the most part, the respondents had very little to say. Now, maybe it's easy. Money, better outcomes, you can say it shortly, but compared to these other questions, both by way of information content and verbiage, actually measuring outcomes was not the strength of the responses. So let's get to it. And I will apologize in advance for going through this talk and just sort of reading my slides. It's not my usual slide, but this is the approach, but this is the nature of this information I'm presenting. So if you will bear with me as I go through the way we've grouped together information, that's how I'm going to do it. So intended applications, 10 institutions. The reason for bringing next-gen sequencing into their institution was cancer. One of those institutions had a CLIA license, and 8 others intended to get a CLIA license. 3 of the institutions, the primary reason was medical genetics. One of them had CLIA certification. And the next question is, so why did you actually embark upon this? And I've actually found this in my own institution as well. Demand from clinical colleagues, shown here in the number of respondents, was a driver in every single institution. Both the clinical stakeholders and certainly the laboratory leadership anticipated that they would have more efficient patient care as well as a more efficient laboratory, as opposed to sending it out to a national reference laboratory and having next-gen sequencing in-house. But these were the 2 primary reasons. And then we get into the remaining answers to these questions are an interesting sociology of why you'd want to have a lab in your institution. Acquire institutional expertise, understand and advance the utility, the clinical applications of personalized medicine. And then we get into something that the reviewers began to react to when I submitted this for publication, which is it's important for our institutional stature, it's important to build research. We need to gain expertise before next-gen sequencing is commoditized and taken over by national reference labs. We want to provide leadership. It is a competitive market advantage. And 2 institutions said we anticipate that it will reduce overall cost of clinical care. To give you some illustration here, I'll bring some comments forward from 2 institutions. This particular institution felt that bringing next-gen sequencing would help them to demonstrate clinical utility, and in so doing would provide a competitive advantage for their institution. They felt that bringing all this material, including the bioinformatics in-house, would be of added value to their clinicians. They certainly felt that it was important to position pathology at the center of the individualization of patient care, personalized medicine, and to be more than just a commodity service where you're reporting values. And they felt that having next-gen sequencing in-house provided strategic advantage for the institution. The comment made by this particular respondent was there's a question of how do you bring next-gen sequencing into the everyday world, when in point of fact, it is still not a low-price test, and certainly when you're standing it up, it's very complicated, and their clinical team, the stakeholders don't appreciate how hard it is to do this in the laboratory, except for sort of the distant view of the clinical practice pathways. For institution Y, again, a value statement. They felt that bringing next-gen sequencing in would enable pathology as a discipline to demonstrate to our clinical colleagues that, number one, this is pathology's domain. We own it. We're going to train people in it. We're going to take care of it. We'll take care of you. And we've got it covered. And also felt that being an early adapter would allow them to get competitive advantage early before everybody else starts doing it. So what are the cons? What are the reasons that you might not want to do it? It's very expensive to bring this stuff in-house. There's a lot of startup cost, and I'll get into that. The time and effort needed to train people, implement, validate, as opposed to just sending it out or using current PCR-based testing is a barrier. From a scientific standpoint, we don't know how to interpret most of the variants in an exome or genome. There are very specific actionable clinical variants, but there's an awful lot of stuff there that we don't know it's significant, which translates into this being complex both technically as well as from a bioinformatics analytical standpoint. And then lastly, curation of the data, both at the immediate time but also long term, has issues technical as well as ethically and legally, and these questions continue to influence decision making. Okay, so barriers to implementing, and these are the institutional laboratory leaders speaking. Scarcity of informatics expertise, the need to develop professional expertise to interpret the findings, rapidly changing technology platforms, the fact that, and you can see these numbers decline. Every institution sort of had its own flavor of what the barriers were. Validating the clinical testing protocols in terms of what's the place of next-gen sequencing tests in management of individual patients. The expense, as I've mentioned, the amount of data to curate, and interestingly, the difficulty in getting out of the gate. A lesson learned is pick your first application carefully. As mentioned, there's an awful lot of information and the results that is not necessarily relevant to the clinical questions, so you get into ethical issues of the reveal. Lack of institutional support for training, and in this instance, this is training of your technical people. You know, the professional staff, the pathology residents is one thing, but this is the failure of the institution to actually allocate the time to train technical people. And then only one institution mentioned uncertainty of the clinical utility, another one uncertainty of reimbursement from payers. So what about bringing this in-house versus outsourcing it? As I mentioned, this is a self-selected group. 12 of the 13 had decided to bring these technologies in. And so a primary reason was control of the pipeline of information, control of the intellectual property that might come out of it, and potentially the ability to retain sort of lean down versions, if you will, test panels within the institution. A fairly common theme was that our respondents felt that sequencing would rapidly become a commodity, and so major labs that can provide this commodity cheaply would be an argument, so that being an early adopter, again, market advantage, gain expertise, potentially retain it in-house because you are adding value to your local practice community. What about the institutional decision-making process? Who pulled the trigger? In one instance, it was a university president, particularly since these activities involved hospital funds. The institutional decision-making officer was the CEO of the hospital, or the COO or the CFO, or in one instance, a hospital capital committee. In only 2 instances was the dean of the College of Medicine the primary decision maker. Cancer center director played a role in one. You can take comfort or not in whether it's the chair of the Department of Pathology that was the primary decision maker. In essence, this is a tale of money. For those chairs who were the decision makers, it's because they were funding this themselves. The same is true for the CEO of hospital, as well as the university people. It's really a story of where the funds are coming from. How is the approval sought to bring this in? In the first instance, the key issues that had to be addressed to justify this is that the laboratory had to define the clinical need. And very important here is aligning your test offerings with institutional expertise. Our respondents made very clear, don't go off offering a test that is not important for your clinical environment. Your priorities should align with the clinical practices and, if relevant, the research practices of your institution. Define the business plan. To me, that's actually hard to do. Number one, if you don't know if you're going to get paid. Number two, if you don't know the ultimate benefit to patient care. But to the extent that you at least have an expensing plan for how you're going to build this and an identified source of funding to pay for it, that's a business plan. You have to put that in front of your institutional leadership. And then, to the best of your ability, define the cost-benefit ratio of bringing next-gen sequencing testing in-house and why you might do that to support existing programs or offset money being spent on send-outs. So, what's the tail of the tape? In 5 institutions, the hospital paid entirely for bringing next-gen sequencing in. In 5, it was entirely the Department of Pathology. One of them was a 50-50 split between the Pathology Department and Cancer Institute. One of them, and this was our Canadian respondent, it's 100% government funding, and the remaining institution had decided not to sink institutional resources into NextGen and was doing outsourcing. They were still practicing genomic medicine, but they were doing it through a reference laboratory arrangement. So now we're going to get into the lessons learned. And this comes from the question of additional institutional requirements. What did you not anticipate that you then had to do? Top of the list was unanticipated on-the-job training of your technologist workforce. Yes, you've got people in the molecular pathology lab. Yes, they're highly skilled, but to bring them up to speed on these machines was the most common unanticipated requirement for additional institutional resources. Not too far behind was the on-the-job training required for your pathologists and PhD clinical scientists, and the need to build the informatics expertise to deal both with the bioinformatics pipeline as well as with the interpretive component of the process. Project management was a key missing element for 2 of the institutions. Basically what they said is, we got our crates, we had our staff, but nothing happened until we actually in one specific instance, petitioned the hospital to also assign a full-time project manager to this. Support from institutional information services was one institutional requirement. And it was interesting to me, in 38,000 words of transcript, that the word governance was only mentioned twice. This was one of them. Quote unquote, the need to establish a multidisciplinary governance process after the fact to drive appropriate implementation of next-gen sequencing testing in their institution. What about reimbursement? And this is very much a dynamic space. So these responses here, yes, they carry forward through 24 months, but I won't claim that this slide gives all the answers. It continues to be true that the laboratory providing the service needs to demonstrate value to payers beyond the current FDA-approved methods, which is in essence an argument that next-gen sequencing panels are of greater cost benefit than single gene tests. And the seasoned people in the reimbursement space, the ones who were already getting reimbursed for this with some modicum of success, said that until the reimbursement hurdle sorts itself out, it's probably best to limit your test menu. So quite honestly, you don't dig too deep a hole. Reimbursement for next-gen sequencing will doubtless follow the rules of the marketplace, but you need to drive it to the lowest price. And, experience would say that conversations with payers can be more productive if you enlist clinical colleagues to be part of the discussion. So, lessons learned. No surprise, it was more complicated and took longer. You need to have a more complete multidisciplinary team, which includes the bioinformatics, the IT and facilities and your clinical stakeholders to really pull this off effectively. Very strong endorsement, not just from these 4 respondents, but also in some of the expanded words. Prioritize proven applications unless you have a strong research environment, which would argue for you're aligning with things that may be less proven in the clinical arena. Play to institutional strengths. Make sure you have adequate informatics support, and capitalize appropriately at the start. Buying incomplete equipment sets, not having appropriate scoping out of the personnel training that you need will stop you in your tracks until you can get the additional resources to do that. It is advantageous, certainly for this one institution, to have a local research partner, and this was the second place where governance showed up, need for a multi-stakeholder advisory group. Okay, a few more examples here regarding lessons learned. Our respondent said that, this particular respondent said small institutions might want to wait for the platforms and informatics and reporting to be worked out, and until clinical utility is more firmly established in the literature, because it's a trade-off. Is it better to be into the field early and be a leader in the field, or wait 2 years, for example, when things are more prepackaged? It's now 2 years later. I'm not sure how well things are prepackaged, but I think the testing panels for cancer, for example, have become more sharply focused, and so this person did have looked down the 2 years pretty successfully. Next-gen sequencing is a much bigger and more complicated endeavor than you would expect, and your success depends on adequate resources at the start. These are not plug-and-play technologies. Do you have the required informatics? It's a whole lot easier to generate the data than to interpret it. And will your clinical institutional workload support your establishing a next-gen lab? Because you're not building a reference lab, you're building a lab to serve your local clinical environment. A second institution, again, important to be adequately capitalized when you start off and staffed. They did feel, this respondent institution felt that you can go from 0 to a customized panel in under a year, and go ahead and embrace the change, but you must work closely with your oncologists, and in this case, hematologists, in other words, a cancer priority, because you don't want to offer something that they're not going to feel is useful. Start simple with a prioritized, focused test offering. Start small, and then go from there. And of course, bioinformatics has to be there. Institution C, this concept of governance. Put together an interdisciplinary group that can help you select the gene panel. This was one of the 2 governance institutions. Getting your feet wet means starting small and build your volume up gradually so you maintain quality and can deal with the huge amounts of data that are coming in. Match your platform with the testing you want to do, and focus gene panel versus whole exome, that debate goes on to this day. But certainly in the state of New York, which is where I am, regulatory issues arise and you need to deal with that. In the clinical workplace, and we encounter that here in the North Shore LIJ health system, your clinical practitioners may want to order individual tests, they may want to order a panel. You just have to work with them to identify the appropriate testing. And then the last institution said, that I'm illustrating, know the politics of your institution and carefully consider the additional work to do a customized panel as opposed to using a commercially available kit. A lot of our respondents said just start with a commercially available kit if you want to get your feet wet. As I mentioned, the 9th question, which is how are you going to measure outcomes, had the least information content. Here it is. 5 institutions said better management of patients. 4 said they're going to measure success by growth in test volume. 2 each, expansion in the test menu and earlier clinical diagnosis. How do you define earlier clinical diagnosis? That's my concern. Achievement of self-sustaining finances. Only 2 institutions said that was a measure of success. And then onesies, which were elimination of other laboratory testing, education research, physician-client satisfaction, patient satisfaction. Number 1, how do you measure it? And number 2, only 1 institution each said that was important. So quite honestly, I look at this and say, we haven't done our job. We have not, in institution, at least at this stage of the game, in bringing next-gen sequencing into an institution, we have not adequately stood up prospective mechanisms to assess success. Research funding, that's one mechanism. I think it's limited to a very select group of academic institutions. Better patient outcomes, it's not on this list. And the commodity that is growth in test volume is a proxy, which may or may not be a measure of better patient outcomes. So I myself challenge all of us who are going to contemplate genomic medicine to be rigorous in how we set up measures of outcomes. That's an editorial comment. I won't apologize for it. So going back to our respondents, what are their expectations and next steps? The expectations of our respondents is that each panel will become easier as you build infrastructure, develop your staff, and improve your reporting. That as clinical trials for targetable genes come through the translational research pipeline, hopefully the panels for next-gen sequencing will become more sharply focused and of more proven utility. And more than one person felt that the exome test, next-gen sequencing, will become a universal assay, so you don't need to do PCR. It's not uniformly held, but it's one way to look at it. The 10th question, recommendations for the College of American Pathologists. You can see the list here, educational programming and so on. But the basic idea was that the CAP definitely could help laboratories in the rigorous parts of standardizing and validating these technologies when you bring them into the lab, and ensuring that your workforce has the appropriate proficiencies and competence. This is my attempt to assemble the flow chart, the workflow for the decision-making process to bring next-gen sequencing into your institution, if indeed that's where you wind up. But this is a flow chart. Identify the clinical need, examine your institutional strengths wherever they may be, clinical research, and very definitely informatics. See whether there are institutional priorities that align with bringing this technology into your institution. Again, clinical research, and this is where market and stature come into play. Establish your business plan, whatever it is, establish it, which can be institutional resourcing, your best guess for reimbursement strategies, and the role of research. Prioritize your clinical applications and then go. Develop your project plan, establish timeline and monitors and your outcomes measures, And I have inserted the word governance here because that, to me, is an important element of project plan development and execution. Yes, obtain institutional approval. Implement, sounds easy, it's a lot of work. Monitor, and then if it's unanticipated resources, you sort of have to go back to the business plan arc, and if you have adequate resources, then you get into this loop of outcomes measures and refinement, in essence, continual process improvement. The 4 pathways that were in play for our institutions are summarized here, and this is a result of the survey, which is why I'm presenting at the end rather than the beginning, is move from PCR-based testing to next-gen testing for the same purposes, and then expand your test menus as you become more familiar with this. Pathway number 2 is just start right out. Do commercial applications right out of the gate for gene panels or whatever you happen to be doing, and then expand. Pathway 3 is the one that was recommended with caution, which is to start out of the gate with customized gene panels and then expand your test menu. And then the last one is just outsource, and then you have to decide whether you're going to do it to industry or academic, and whether your informatics are going to reside with industry or you're going to have them in-house. But in the end, you're going to be looking at questions of whether you're doing a very small targeted panel of genes or expanding it to 100 and beyond, and then ultimately what's the role of exome sequencing just becoming one more test. Subsequent to our actually doing the interviews, a National Cancer Institute investigator team published in Genetics and Medicine Cancer Institute directors' responses to a survey of the difficulties in practicing genomic medicine. And these are their responses. For Cancer Institute clinical leadership, their concerns with next-gen sequencing were the amount of data that has to be curated from these lab tests, the fact that it's difficult to catalog clinically actionable variants, and there's limited consensus on what these variants mean. For them, a concern was lack of reimbursement for the clinical time it takes to practice genomic medicine, leading to a burden to providers in many ways, one of which is following up on the clinical interventions that you plan on doing. So these are the clinical issues that were raised in this paper over and above the ones that we revealed in our survey. Put them together, and on the upside, these are the key concepts for all of our respondents, which is opportunity to advance patient care, for them imperatives for developing knowledge and expertise in this field and developing an evidence base for next-gen sequencing use in the long run, the need for these institutions to provide leadership, and yes, institutional prestige. Okay, now comes the fun part. How did this survey fare when I submitted it for publication? And this is a distillation of the peer review comments that came out of the review. Two reviewers, this is a synthesis of what they said. This study does not touch on the issue of the appropriateness of the implementation of next-gen sequencing, which makes discussion of implementing next-gen sequencing difficult. The authors should comment on whether a desire for recognition is a good reason to do this, and the broader issues of implementing these technologies, namely clinical practice especially, are only tangentially addressed, so that this is the nub of the issue, and I'll come back to this in a moment. Reviewers felt we needed more information on the business case because it was quite weak, to be honest with you, and I agree with them. And we need more information on how this technology might create problems in patient care as opposed to solve problems. But the fundamental issue with this study, with which I agree, is this study does not answer the question of clinical utility. And in the reviewer asking of us, well, what's being done to assess clinical utility? I've given you the answers. This is you've seen the responses of the institutions, which was what, shorter time to diagnosis. No comment was made about a way to establish metrics for improved patient outcomes. So the two reviewers were concerned that the technology was not necessarily driving more efficient care or better outcomes. In my response to the reviewers, I said basically this is the issue. A decision has been made without knowing the clinical utility. And I agree with you, but that was not actually the purpose of the study. The purpose of the study was to examine the decision-making process such as it was. An editorial was published on the same issue of Genetics and Medicine, this being July of 2014, where the authors said our study is off target because we should be looking at utility before business. So to summarize the editorial, whatever the rationale for the study, the survey neglected to first assess central and difficult issues and why these leaders' perceptions of the clinical utility of next-gen sequencing was valid. Was it to be perceived as scientific leaders, then this should be a research tool. Was it to be perceived as a market leader, then you're doing this without sufficient concern about utility or costs. And is it simply because it's now cheaper? You haven't demonstrated that it's either medically or financially advantageous, so that analytical validity and clinical validity are necessary but not sufficient conditions for use, and without demonstrated utility, the potential for waste and harms outweighs hypothetical benefits. So they summarized by saying, evaluations such as this one have great potential to inform the thoughtful introduction of whole genome sequencing, but you need to ask and answer the right questions, not just the practical and business ones. And I read this with a smile because they're getting at exactly the issue, which is there's tremendous momentum now for performing next-gen sequencing. And the medical science is spectacular, but the clinical utility has not been established in a systematic fashion. And we have to make decisions as to whether to perform this testing and to deploy it within our institutional environments without sufficient knowledge of whether it's the right thing to do. So I wind up concluding that other than dramatic case reports and series, even 2 years on from when these respondents answered their questions, the clinical utility of next-gen sequencing has not yet been demonstrated. But there are, in the eyes of these respondents, compelling reasons for institutions to decide to implement next-gen sequencing in genomic medicine. Our study examined the decision-making process, not whether these were the right decisions to make. And my closing thought is that only careful future study can answer the question of clinical utility. This is debated within my own institution, which is, do we need to have randomized clinical trials to answer that question? And I can't answer that question in this talk, but I leave it for you to contemplate. So this is my email if you want to carry on dialogue after our discussion. But at this point, I will close, and we can turn to the question and answer period. And I believe Paul will moderate, but I can also take a look at questions as well. So I will end, and Paul, turn it back over to you, and you can put questions into play.
Thank you so much, Dr. Crawford. Very, very interesting presentation. And I think I would say, both in my capacity as a board member of Gnome, but also as an investor in the space, many of the issues that you highlight here are very consistent with the questions that I think are being broadly asked by stakeholders, throughout the continuum around questions around clinical utility and what does what is the process via which people choose to adopt. So very, very insightful, and greatly appreciate the talk today. I do want to turn to a few of the questions that have come up over the course of your presentation, and I will say a couple things before we dive into the questions. First of all, I neglected to mention at the beginning of this presentation that we had more than 450 people registered for today's event. So I think that that's illustrative of the level of interest in the topic of discussion. I guess the first question I'd like to start off with, and I recognize you may not feel qualified to answer it, but I will ask anyway. Based upon what you've seen in your research, and also what you've seen other institutions do, and what you yourself are participating in, at North Shore LIJ, what would you say is a prototype for good governance? What would good governance look like to you?
Well, I mean, thank you for asking that. Oh, you had me worried there for a moment. So yes, I've emphasized how strongly I feel about governance, and hopefully you'll detect that in the answer. Let's start with patient care. The medical science that is so spectacularly coming into play through these technologies ultimately, hopefully, will improve the human condition. And and I'm speaking from my heart. The applications are in front of us with regards to cancer genomics, somatic genomics, pharmacogenomics, and notice my exclusion of microbiomics. That sort of falls into a different sector. And so, the dream come true would be that through identification of the molecular makeup, and right now it's next-gen sequencing, I'm going to stay away from epigenomics and that sort of stuff, that we will be able to identify clinically actionable genetic variants to the benefit of the patient early in their course, and in so doing have more rapid time to diagnosis, have more effective response to therapy, and yes, more efficient delivery of healthcare. What is the governance it takes to achieve that? It starts with your clinical stakeholders. And I've been 6 years in the North Shore LIJ Health System, and both the Department of Pathology and our Cancer Institute and maternal fetal medicine and pediatrics, we were all traveling our journeys, and my feeling was, quite honestly, in the year 2014 is when our stakeholder groups had reached the point of maturity so that we could bring together the governance to go through what is now very deep into our decision-making process as to the future role of genomic medicine in this health system. And so the stakeholder, for me, and others can look at their own institutional environments, the stakeholder groups are cancer, pediatrics, OBGYN writ large. It boils down to maternal fetal medicine, research, and that is the fourth of our five stakeholders, and pathology and laboratory. But sort of overarching all of that is the institutional corporate governance, whatever the structure is, for business management, and how you are going to, and yes, I should mention also information services. So I've named 6 stakeholder groups that are involved, and the nature, I mean, the manner in which they come together is going to be unique for any given setting. I'm speaking from the position of an integrated health system, but there are so many different models of governance and business architecture of regional healthcare delivery systems. So for example, if you're in an independent, a local or regional independent pathology group, either resident within a health system or separate from a health system, that's going to dictate a different form of governance. But one way or the other, I would say start with your clinical stakeholders and go from there.
That's a great answer, Dr. Crawford. And I would just amplify that by saying, from a business side of the equation, as representing, as I said, as a company who's trying to approach institutions about offering technologies to solve some of these problems, there is clearly a difference between an institution who has a good governance process that involves many of the different stakeholders, whether it be pathology, molecular genetics, oncology, whoever is relevant, and an institution who doesn't have that kind of governance in their ability to effectively decide what they want to implement and how quickly they can go. So I certainly can agree with what you're saying about the role of governance and how that, while it needs to be tailored to each institution, that's very consistent with what we've seen.
Okay, before you start your next question, let me just interject.
Go ahead.
One of the questions that I see on my screen is, I'm looking at the July 2014 issue of Genetics and Medicine, and I'm not seeing the paper, the editorial. I've kind of been wondering myself. I just went on PubMed myself, and it still is listed as EPUB before publication, and that's true for both the editorial as well as our paper. It's an open. We published it as open access, so you should be able to download either way. If you want me to send you PDFs of both papers, I can do so. There's my email, if anybody's having trouble getting a hold of this. So anyway, back to you, Paul.
Oh, no problem. So Dr. Crawford, another question that came up very early on was the, well, what should we do question? And I think that's the question that a lot of people are curious to answer. And I know that you can't answer that question, but I was very intrigued.
What's the right answer?
What is the right answer? But I was very intrigued by the 4 pathways that you laid out. I think it was your slide on page 36. My question is, would you be able to give your impressions as to for what kinds of institutions would one pathway versus the other be more likely to make sense?
Okay, this is where I'll rapidly get into deeper water with my own, if you will, administrative expertise, but not a deep expertise in molecular pathology per se. Let's say you're doing PCR-based testing for the CFTR gene, and you want to move into next-gen sequencing for the CFTR gene. That would be an example of sort of a single application, single migration activity. And I'm sure there are others that speakers could comment on much more with a much greater level of expertise. Pathway 2 is the one that really was the sweet spot for this entire discussion, which is, fine, you've got a PCR-based testing laboratory, that's great, but just sort of start fresh, whether it's lung cancer or colon cancer or now melanoma cancer, forget my vernacular here. Choose your application, identify the next-gen sequencing panel. Again, we're talking about insourcing here, not using an industry reference laboratory. Choose the application, what is the panel, set it up, make sure it's aligned with your clinical stakeholder groups. And having done that, then expand your test menu into other clinical application areas. So really, in this entire discussion, I would say that's the sweet spot. The argument for pursuing a custom next-gen sequencing panel really is a local strength argument. Let's say you've got a group that is a world leader in cardiomyopathy or something like that. That would argue that you work specifically with both the clinical and the research enterprise and design a custom next-gen sequencing panel. But this is not for the faint of heart. And quite honestly, as our health system is going through its decision-making process, I will tell you, this is where we're landing. We are looking at an outsourcing model, and for us, the dividing line is not the technical, because we're going the industry route or an academic partner. This is exactly where we are right now in our decision-making process. But in the sort of hanging in the balance is how much informatics do we build in-house as opposed to keep with our partner. But this is actually the pathway that the North Charlotte J Health System is traveling as I speak.
Thank you. No, very, very helpful and very clear. I was also struck by one other thing that came across in your presentation, and there were a couple questions relating to this. That the barrier seems to, at least as these institutions reported to you, and by definition they are early adopters, but they seem to see the barriers more around expertise than as cost. And obviously
you
those
noticed.
are intrinsically linked, right? Because you got to buy expertise often, and you have to train to create expertise.
Yeah.
But I'm curious as to just any insights that you had about that. Was that surprising to you, and do you think that that's going to be true going forward?
It's interesting because one of the respondents, when asked about how did you go through a decision-making process, the respondent said, "Well, you just convinced them." That's an interesting approach to a business plan. If an institution decides, for whatever reasons, and I've given you plenty of them, that it's important to own this technology and to have it in-house, then it becomes an internal decision-making process as to how the institution will resource that. But I think it's fair to say, certainly in 2012, the revenue was not there to support the build. You're doing it on institutional funds. Certainly 2 years ago when we did the survey, it was hard to build a business plan saying we're going to, as an institution, make money on it. 2 years down the line, there's been tremendous activity in the reimbursement space. And as we look at it as our own institution, again, trying to anticipate a clinical revenue cycle which justifies a build is a challenge. I'm not saying it can't be done, but you've got to have some sense of stopgap or stop loss for how you're going to deal with that. This gets into a very interesting topic, which I didn't address at all, which is, well, how does this whole business argument change when you get into 100% capitated risk? And to me, that's the end game, and it's an end game that I think will really help answer this question, because reimbursement becomes moot. It truly becomes a matter of whether it's smarter medicine from a cost-benefit analysis to do next-gen sequencing early and do it in a comprehensive fashion. And that's why I think these next few years are so critically important for many institutions, early adopters as well as mid-adopters, to actually answer the question of whether it's better, because that will guide the accountability, accountable care if you want to call it that, of doing next-gen sequencing. To me, the end game is it's smarter to do the testing. We get better outcomes. In these clinical settings, let's do the testing, and it's part of the total spend on care. We're not there yet, but for me in 2014, the reason in the beginning of 2014 I felt it was time for our health system to enter into the fray is that I feel that the time has come for us to help answer that question from a clinical informatics and patient outcomes standpoint.
Makes sense. And, it's such a fast-paced and fast-moving area. I mean, I think it was only 2 months ago there were announcements from both Illumina and Thermo Fisher, amongst others, about partnerships with, some of the leading pharmaceutical companies, Pfizer, J&J, GlaxoSmithKline, about sort of establishing, if you will, the right panels for next generation.
Right, and then for all of you listening in, you'll notice that I've assiduously stayed away from any comment on the technical platforms. This is the medical science as it maps into the clinical and business space.
Understood. And I think, I guess where I was going with that is that just as new drugs are being developed and launched all the time, the fact that treatments are available fundamentally changes the value proposition of this testing, right? Because once there's a drug,
well, that's to a certain extent, those are the two closest dots. Have actionable target, you can target it. But we're dealing with broader things which include diagnosis, not just actionable targets.
That's right. That's right. But I think the incentives that will move the adoption forward. It's very clear from your research that there's just an overwhelming drive coming, particularly from oncology as well as other clinical applications as well. And I think that momentum is probably going to be maintained. Well, unfortunately, I think we're about out of time. We've reached the top of the hour. What I will say, though, however, is that this seminar, this webinar, has been recorded and will be available for download on the Gnome website for people to have access to. And in addition, we will also be providing Dr. Crawford with all of the questions that we didn't have time to cover today, and so he'll have the opportunity to get back to them personally via email if there were some that we didn't have time to cover. Again, Dr. Crawford, I would love to thank you for taking the time to present your research with us. Very insightful, very interesting, clearly very topical. So thank you very much.
Okay, thank you.